Dear students, now we are going to see diagnostic agents. So what are diagnostic agents? So these are the substances which are used to examine the body, which are used to diagnose the body. Why we have to diagnose that? For detecting impairments of its normal function. So these are the substances which are used to examine the body for detecting impairments of its normal function. That's why they are defined as they are the chemicals or substances which are used to detect abnormalities in tissues or organs or to test an organ function. So this is mainly used for diagnostic purpose and these are not having any pharmacological effect. These diagnostic agents are classified on the basis of the test functionalities into 12 types. So first one is gallbladder function. So the examples are iositamic acid, iodipamide and thyropanagate sodium. So second type is gastric function. Examples are pentagastrin and Congo red. Then third one is liver function. Example is indocyanin green. So next one is ophthalmic diagnostic aids. Example is fluorescent sodium. Then fifth one is pancreatic function. Example is bentiromide. Then sixth one is intestinal function. Examples are barium sulfate and xylose. Then next one is kidney function. So examples are aminohipurate sodium, indigotin disulfonate and phenol sulfothalin. So next one is lymphatic system. Example is isosulfan blue. Ninth one is bronchial airway hyperacidity. Example is methacholine chloride. Then 10th one is drug hypersensitivity. Example is benzyl penicilloyl polylysine. So next type is drugs used in X-ray contrast medium. Examples are diatriazoic acid, iositamic acid, iothalamic acid, propyl iodone and iodipamide. Last type is miscellaneous. Examples are erythrosine sodium and Evans blue. So totally 12 types are there. Now we'll see the first one, that is called as iositamic acid. So look at the structure of iositamic acid. It is having phenyl ring, that is having 3 iodines with 2 amino groups. This nature of this amine is primary, whereas this one is tertiary. So this tertiary nitrogen is connected to acetyl. Then this tertiary nitrogen is also connected to methyl. That methyl is connected to propanoic acid. So what is iositamic acid? We have the phenyl with 3 iodines, 2 amines. One is primary, another one is tertiary. Tertiary is connected to acetyl, then with methyl. That methyl is connected to propanoic acid. For clear understanding, we are dividing the word iositamic acid into iositamic acid. Io is indicating that 3 iodines and cit is for the acetyl, indicating the acetyl, and am is indicating this tertiary nitrogen or tertiary amine, and IC acid is indicating propanoic acid. So IO for iodine, cit for acetyl, am for this amine, and IC acid is for propanoic acid. And these are used as an oral cholecystographic agent for radiographic visualization of the gallbladder. So what is the use of iositamic acid? Mainly used to diagnose the gallbladder. Next one is called as thyropanagate sodium. So look at the structure of thyropanagate sodium. So this is also having a phenyl with 3 iodines but with 2 more substituents. One is an amide, CONH is a, that's why it is an amide with 4 carbons. That's why it is a butanamide. And look at the other end. This end is having a propanoate, that is 3 carbon ester. 3 carbon acid is propanoic acid. So this is an ester, that's why it is a propanoate. For understanding, we are dividing the word thyropanate into thyro and panate. So thyro is indicating the presence of the amino acid called as tyrosine. So if it is an amino acid, center one carbon is there that is connected to 4 grooves. In the 2 are mandatory, that is amino acid. Fourth one is H. In the fourth substituent only, sorry, third one is H and fourth one no. So in the fourth substituent may vary for all the amino acids. So here, what is the fourth one for tyrosine? That is methyl with the para-hydroxyphenyl. What is tyrosine? Center carbon, amino acid, one H. Another one is methyl with the para-hydroxyphenyl. So this is tyrosine. And for panate, panate is indicating that propanoate. So what is thyropanate? That is having tyrosine and also propanoate. Now compare tyrosine with this, compare the aromatic ring. So here both are having the same aromatic ring, that is the benzene ring. But here it is having hydroxy group at para position, whereas here that was absent. So instead of the hydroxy, it is having 4 groups, 4 substituents. So 3 iodines with 1 butanamide. Another change is this amino group, no, so that is replaced by means of ethyl. And it is a sodium salt. That's why this H is replaced by means of Na. So it is used as a oral iodinated contrast agent that is used for radiographic imaging of the gallbladder by means of the procedure called as cholecystography. So 2 agents we have seen. So third one is diatriazoic acid. So look at the structure of diatriazoic acid. This is also having a phenyl with 3 iodines. So here, what is the parent? The parent is benzoic acid, that is benzene ring with acid, benzoic acid with 3 iodines with 2 acetamide groups. So we have 2 acetamides. What is diatriazoic acid? That is benzoic acid with 3 iodines with 2 acetamide. For clear understanding, we are dividing the diatriazoic acid into diatriazoic acid. So now take dia-A. Dia is indicating 2. 2A. So what are the 2 A's here? One is 2 acetamides we have. So dia-A is for 2 acetamides. And tri is indicating the 3 iodines. Zoic acid is indicating the benzoic acid. So we have a benzoic acid with 3 iodines. Tri. Zoic acid is benzoic acid. Tri for 3 iodines. And dia-A is 2 acetamides. This one is otherwise called as amidotriazoic acid, trizoic acid, and it is an iodinated X-ray contrast agent used in medical imaging to improve the visibility of the internal organs and blood vessels during X-rays and CT scans. Next one is called as Congo red. So look at the structure of Congo red. This is having the parent as benzidine. So this is benzidine. What is benzidine? 2 anilines were fused. Both were connected at the para ends. Both the para ends were connected means that is called as benzidine. So now that benzidine nitrogen, no, that is connected to one more nitrogen. How these are connected? By means of a double bond. That nitrogen is connected to the naphthalines. Both the ends that were connected to the naphthalines. So the amino naphthalene. So here parent is having NH2 here. This is also having NH2. From both the NH2, 2 hydrogens were removed. That's why both are connected by means of a double bond. So we have a benzidine. That nitrogen is connected to one more nitrogen by means of a double bond. That one is connected with the naphthalene. Look at the naphthalene. On upper ends, both the naphthalines were having amino group, and the lower ends they are having sodium salt or sulfonic acid. So what is Congo red? Benzidine is there. That is connected with one more nitrogen by means of a double bond, and that is connected with naphthalene. Upper ends are having NH2 on both the ends. Lower ends are having sodium salt or sulfonic acid. This is Congo red. So it is a benzidine based azodine used mainly as a histological stain in pathology for detecting amyloid proteins in the tissues. So next one is Evans blue. So look at the structure of Evans blue. So while comparing with the Congo-red, it is having similar structure with some variations. So here center benzidine is there, that is having 2 extra methyls. These 2 methyls were extra, and actually this end is having amino group. So that is moved here. Amino present here is moved here. That's why this place is having O hydroxy groups. And this end is having sodium salt or sulfonic acid, that is moving here. And extra sodium salt or sulfonic acid, that is Evans blue. So how many changes? 4 changes. Extra 2 methyls and amino and sodium salt or sulfonic acid. Actually they are present in these 2 positions. They are moving to these 2 ends. And this is occupied by hydroxy groups and extra sodium salt of 1 more sodium salt or sulfonic acid. So that is Evans blue. So it is used to check the vascular permeability for detecting blood vessel damage by binding to the serum albumin, and also leakage from vessels, that is used to diagnose the leakage from the vessels into the surrounding tissues. It is used to detect blood-brain barrier breakdown in neurological disorders, identifying the tumor lesions and also localizing lymph nodes. So next one is indocyanin green. Indocyanin is a tricarbocyanin dye. So what it is? It is a tricarbocyanin dye. So first we have to know what is a cyanin dye. Actually dye, it is a synthetic dye and that is having 2 heterocyclic rings. That is 2 heterocyclic rings and both the rings are having nitrogen. So it is having 2 nitrogen containing heterocyclic rings and that 2 rings are connected by means of a conjugated polymethane chain. So that is connected by means of a chain that is having conjugated double bonds and also a polymethane. So it is having many carbons. So many methyls are there but with the conjugated system. So that is cyanin dye. So next one is carbocyanin. So same heterocyclic rings with the nitrogen, but change is it is having 3 carbons, that is conjugated chain of 3 carbon atoms. So this is polycarbons. Polycarbons are there, that is called as cyanin. 3 carbons are there, that is called as carbocyanin. So next one is tricarbocyanin. Here 7 carbons are there means that is called as tricarbocyanin. So polycarbons are there means carbocyanin. 3 carbons carbocyanin, 7 carbons that is called as tricarbocyanin. Indocyanin is a tricarbocyanin dye. So now we'll see the structure of this indocyanin green. So already what we have seen, center is having 7 carbons with conjugated system. So look at this. So this is the linkage that is having 7 carbons. 1, 2, 3, 4, 5, 6, 7. 7 carbons. That's why hepta. Conjugated. Single bond, double bond, single bond, double, single, double, single, double. So conjugated system. How many double bonds are there? 1, 2, 3. So within the 7 carbons, it is having 3 double bonds. That's why it is a heptatriene. So linkage is a heptatriene. We should have 2 heterocyclic rings and both should have nitrogen. Only this ring is called as pyrrole. Fusion of pyrrole with benzene, that is called as indole. That indole is fused with one more benzene. That one is called as benzoindole. So heptatriene is connected to both the ends by means of the benzoindole. That benzoindole is having 2 methyls. 2 methyls are there. Lower end, it is connected with, this is sulfone group. SO2 is sulfone. This is butyl. So that too were sulfobutyl group. So what is indocyanin green? It is a tricarbocyanin dye. That's why 2 heterocyclic rings with the nitrogen connected by means of 7 carbon chain with conjugated system. So heptatriene in the center. Both the ends were connected with the benzoindole and that indoles are having 2 methyls in the upper end. Lower nitrogen is connected with the 2 sulfobutyl groups. It is a sodium salt. So it is having 2 benzoindole moieties are connected by means of a heptatriene chain. So it is having sulfobutyl groups, no, where they are in the indole rings. So because of that it is water soluble. So next one is fluorescent. So what is fluorescent? Actually it is having fusion of three rings, three 6-membered rings. So a fluorescent is a xanthine ring system. This is having xanthine ring system. So this upper NO, that is the xanthine ring system with the spirolactone. So this is a lactone, but this is a spirolactone. Because two things are sharing the same carbon means that is called as a spiro one. So now we'll see the structure of this xanthine. So what is xanthine? It is the fusion of three rings. First and third are benzene rings, and center one is a pyrene ring. So fusion of these three is called as xanthine. So now compare this one with the fluorescent. So this is a xanthine, but how many? Actually xanthine is having 6 bonds, whereas look at this, here also 6 bonds, but the position of bonds would be varied. So first one is having 3 bonds, second one, third is having 2, and this is a lactone. We said no, that's why we have a ketone there. Extra groups are one hydroxy group, and the center position that is connected to the second position of benzoic acid. So it is having a xanthine ring system with the spirolactone, and the center one is connected with the benzoic acid, and this is also connected with one hydroxy group. So this is for fluorescent. For fluorescent sodium, these two hydrogens are replaced by means of Na. So that is called as fluorescent sodium. So what is fluorescent sodium? It is a fluorescent organic dye mainly used as a diagnostic tool in ophthalmology for detecting corneal lesions and abrasions and also ulcers. In fluorescent angiography, it is used to diagnose the retinal and intraocular diseases. It works by glowing yellow-green under blue light and highlighting the damaged tissues. They can be applied topically to the eye or injected intravenously as a fluorescent tracer to study the vascular permeability and also the cellular properties. Next one is erythrosine sodium. So look at the structure of erythrosine sodium. It is having structural similarity with the fluorescent sodium. So while comparing with the fluorescent sodium, it is having 4 iodines. That's all. So only changes extra, it is having 4 iodines. That's why it is a tetraiodinated xanthine dye, that is a fluorescent dye, or we can write it as it is a tetraiodinated fluorescent sodium. And this one is otherwise called as xanthine, already we have seen. So this one is otherwise called as red number 3 or E127. It is a cherry red synthetic dye mainly used as a food coloring, biological stain and dental plaque disclosing agent. So next one is bentriomide. So this is, look at the structure of bentiromide. So this would be the structure, and this one is otherwise called as BT-PABA. What is B? That is benzoyl. T is for tyrosyl. PABA, we all have known this already while seeing sulfonamides, that is para-amino benzoic acid, otherwise called as Chimex. So benzoyl, tyrosyl, PABA. Where benzoyl is, that is connected to the nitrogen, that's why N-benzoyl. Usually amino acids are existing in two configurations. One is capital D, another one is capital L. So here what is the configuration of this tyrosine? That is L. So what are the parts present in this agent? That is benzoyl group, tyrosyl group and PABA group. So what is tyrosine? Already we have seen it is an amino acid. So center carbon with the 2 mandatory groups, amino and acid. Third one is H. And what about the fourth one? Methyl parahydroxyphenyl. This is tyrosine. So look at this. So here we have a tyrosine. So one part is tyrosine, and that tyrosine is connected to the PABA. PABA means para-amino benzoic acid. Para-amino, this is benzoic acid. How tyrosine and PABA were united? The acid of tyrosine reacts with the amino group of PABA. Acid plus amine means there may be removal of one molecule of water, we are getting an amide. So tyrosine acid and amino group of PABA combines and forms an amide linkage here. So what would be the left one? Left is benzoyl group, where benzoyl is added with the nitrogen, with the N. That's why that is called as N-benzoyl, L-tyrosyl, para-amino benzoic acid. What are the three parts? Tyrosine part. Acid is connected to the amino group of PABA, forming an amide linkage, and in this the amino group of tyrosine is connected to the benzoyl group. So what is the other name? BT PABA, that is N-benzoyl, L-tyrosyl para-amino benzoic acid. So it is a synthetic peptide formerly used as a screening test for exocrine pancreatic insufficiency, and also to assess the function of the pancreas by measuring the activity of the pancreatic enzyme called as chymotrypsin. Now it is withdrawn from the market. So next agent is aminohippurate sodium. So look at the structure. So we have the structure of hippuric acid. Hippuric acid means that is having two parts. One is this part is called as benzamide, and that benzamide is fused with acetic acid, means that is called as hippuric acid. What are the two parts of hippuric acid? Benzamide with acetic acid. This is aminohippurate sodium. So amino group is added to the para position, and it is a sodium salt. That's why H is replaced by means of Na. So what are the two parts of hippuric acid? Benzamide with acetic acid. This is otherwise called as para-aminohippuric acid, or PAH. It is used intravenously to measure the effective renal plasma flow and also to check or assess the kidney function by evaluating the kidney's ability to filter and secrete the substances. And last one, that is called as phenol sulfothalin. So look at the structure of phenol sulfothalin. So here this is the basic nucleus present here. We all have known this is benzene that is fused with one heterocyclic ring, that is a 5-membered one with oxygen and sulfur. So in that, oxygen is getting higher priority, OSN, that is the order of priority. So this is number 1, this is number 2. So that's why this is called as benzoxathiol. So what is the nucleus present? That is benzoxathiol nucleus. And that sulfur is connected to oxygens. That's why it is called as dioxo benzoxathiol. So which would be the numbering? This is number 1, 2, 3, 4, 5, 6, and this corner is 7. So that 7th position is connected to 2-para-hydroxyphenyl. So that is called as phenol sulfothalin. So what is that? It is having dioxo benzoxathiol, and 7th position is connected to the para position of 2-para-hydroxyphenols. Para position of 2 phenols or 2-para-hydroxyphenols. So this is otherwise called as phenol red, and this is used as a pH indicator in cell culture and laboratories. It is used as a diagnostic aid to diagnose the kidney function by measuring the excretion in urine. So that's all about diagnostic agents. Thank you.